Hello, everyone. Thank you so much for having me. Um, I am beaming in from Sydney today. We are under a deluge of rain, specifically from Darawal country. So today it's a pleasure to be here. I wish I could be there with you. I am a Victorian girl, but I've been up in Sydney for about 12 years now. Because that's where the mites are currently. So that's where I can do all my research. Okay, let me share my screen and I will get started with my presentation. Let's see. Now, can you see my slides there? We're good? Good to go? Awesome, thank you. Okay, so today I'm going to talk to you about some of my research into honeybee viruses. And yeah, I've been working with bees for about 12 years now, and for viruses for most of that time. And I had also been working a little bit on this critter, varroa mite, prior to its arrival in Australia because its biology and its ability to transmit viruses is something that I've always been pretty fascinated by. So today I'm going to focus on viruses. This is my group that I work alongside at Sydney University. We have heaps of fantastic students that come through our lab, learn how to be beekeepers. My co-lab lead is Ros Glokia, and I also have worked very closely with Nadine Chapman. And our previous lab heads, you may have seen some of these in your past conferences, Ben Oldroyd and Madalena Beekman. So we have a great group of people there that we work with, and we have some bees on campus. We've been slowly awaiting the arrival of Varroa, and it has been in our hives now for about 9 months. Our primary form of varroa control has been students taking mites out of the comb because they're so excited to experiment with them. So we've barely had to use any chemicals at this stage, but they're slowly starting to ramp up now. So we are exceeding even my students' ability to keep the mite levels low. So the three areas that most of my personal area of research covers can fall under these three P's of bee health: pathogens, so the viruses, parasites, that's varroa mite, and also pesticides. So I do also do a bit of work into understanding pesticides and how we can prevent negative impacts of those. So for today, I'm going to focus on the pathogens, the viruses, and sort of my goal overall is to understand how new pests and diseases will impact honeybees, and then take that information and figure out new methods to improve the health of bees. So, Varroa and honeybee viruses are one of the major causes of suboptimal honeybee health globally. And with the arrival of Varroa in Australia, the big question on everyone's lips is how it's going to impact our Australian viruses. So I'm going to go through a few sort of ideas and scenarios today, and a bit of my background from work overseas, and see how we can then translate that into Australian messages. So viruses, I think in this COVID era, people know a lot more about viruses than what they may have in the past. So we have these tiny microscopic particles that are very abundant. They infect every organism that we've looked in. They evolve really quickly, and they are incredibly diverse. So they are all these tiny little structures that can infect huge ranges of organisms. Basically, the general idea is the virus cannot live on its own. It needs a cell or a host for it to survive, but it can sort of enter that cell, and then it uses the machinery of the cell to replicate itself, and then it will cause that cell to lyse, and then viral particles can then spread throughout other individuals that can be infected by that virus. Now, it's important to know what a virus structure is for my talk. So I'm going to go through a little bit of that. Basically, a virus contains this sort of capsid protein-based shell, basically like the outer shell. And then on the inside, it has the genome. So like all organisms, viruses have genomes. It's basically that sequence of nucleotides that tell the virus what to do. And in viruses, they are very diverse in terms of what their genomes look like. So their genome could be made of two different molecules. so ribonucleic acid or deoxyribonucleic acid. Our genome is made out of this one, DNA, so deoxyribonucleic acid. But most of the viruses in honeybees actually have an RNA genome. And it's this genome that's really fascinating because it mutates very rapidly, and so we get lots of diversity. Now, not only is the ingredients different, but it's also different shapes and sizes of the viral genomes. So we can have an RNA genome that we call a negative sense RNA virus. Some examples that you might have heard of before. So the flu, it's a negative sense RNA virus. We can also have positive sense RNA viruses. COVID is a key example of one of those. And then on the DNA side, we can have two types of DNA viruses, double-stranded or single-stranded. Now, we don't tend to have those in bees, so I'm not going to really talk about them anymore. But just so you know, that's the sort of makeup of most viruses in the entire sort of animal kingdom and plants. So bees, as I said, mostly have these two types of RNA ones. Now, historically, there's been some very common honeybee viruses that we've known about for hundreds of years. These are the ones that cause very obvious symptoms. So we have things like black queen cell virus and sackbrood virus. Australian bees have commonly got these viruses. And throughout the world, they are also the most common viruses. So we have black queen cell, sackbrood, and then we also have some other viruses that are really quite common throughout the world and Australia, like Lake Sinai virus and paralysis viruses like Israeli acute. But then there are some viruses that we don't yet have here, sackbrood and, slow bee paralysis and deformed wing virus. Now, these are the viruses that people frequently test for when they're looking for diseases in their colonies, because they're the most common, they're the most ones that we know symptoms for and such things. We know them to be bad for bees. But we have had a huge influx of new methods in the last few years, which has allowed us to discover many new viruses. And so I became very interested in this novel virus discovery methods and decided I would test this out in some samples that I'd received and gone and collected throughout the world. So just to reiterate about the honeybee viruses, they're mostly from these two families of viruses. So this is a family tree. If you think of your own family tree, basically it shows how related you are to the individuals by connecting you via lines. And it's basically the same thing for a virus family tree. We have these two families of viruses, the Iflaviridae, Dicystroviridae, and most of our common bee viruses fall within these families. And so I've just pointed out a few of the key ones here. They're all positive stranded RNA viruses. So, as I mentioned, I was keen to use this new virus discovery method. It's called metatranscriptomics. And the general idea, I don't want to go into too much detail. It is, after all, 9 o'clock, and you probably just had your first coffee for the day. So maybe this would be a conversation for the pub afterwards. But to give you a brief overview, we sample bees from all over. So this is me in Tonga sampling bees in 2015. I also had lots of helpers sampling from other places throughout the world. And then you basically take the RNA from the bees. So you use some molecular methods to extract RNA from the bees, and we sequence that RNA. We basically sequence it in tiny fragments, and then we put those pieces back together to make virus genomes. Oh, sorry, that's just ruined everything. Okay, and then we search our database for sequences that actually match to other viruses from other places. And so we're basically searching for this nucleic acid, this sort of genome part that we've extracted, and we're looking for any sort of very distant matches to any known viruses to try and figure out which ones are new viruses in the bees. So when we sequence all of the RNA inside the bee, basically it involves grinding up 20 or so bees from a colony. We take the RNA portion of that bee goo. It's basically like a bee smoothie, which you extract the RNA from. And then you send the sample along with a bucket load of cash to a sequencing company. And they will sequence every single RNA piece inside that bee, including all of the things that are in the bee. So you're getting the bee's genes, you're getting any parasites that are in the bee, you're getting any gut bacteria, and of course you're getting any viruses that are inside the cells of the bee as well. So the most challenging step of this is to actually piece those reads back together at the other end. So you sequence it, you put the pieces back together, and then you look for novel viruses from those. Okay, so the main finding from my research was that I found these two new viruses called Apis rhabdovirus 1 and 2. So these were found in samples that I'd collected from Europe, so the Netherlands, Tonga, New Zealand, and South Africa. And others have since found them throughout the US, Israel, and now looking into the literature, this was about 8 years ago now, we're finding them everywhere pretty much. So everywhere where Varroa is has been examples of finding these rhabdoviruses in bees. So these ones are interesting because they are different to every other bee virus that we found. They have, instead of a positive sense RNA genome, they have a negative sense RNA genome. Now, the other key factor, which will come into play, is that they are present in bees and mites. So they're an example of a virus that can replicate in both the bee and the mite. And this is actually not that common. So having a virus that can replicate into quite different hosts. So there'll be more on rhabdoviruses shortly. So before I move on, these are two rhabdoviruses that form quite a distant relationship with each other. So that while they're both rhabdoviruses, they're not like neighbors, they're not very closely related to each other. They're called rhabdoviruses because their particles have these rod-like structures, and this is their negative sense RNA genome that gets coiled inside the capsid. They have 5 genes, so very few genes. When you think about a human genome, we have thousands of genes, and these viruses only have 5. The interesting observation is that they were present in every mite that we looked at, but they weren't present in every bee. So it looks like it's a virus that just comes along with the mite package. If you have a mite, the mites are going to have these viruses. And when we looked at their immune response to viruses, now I won't go into any detail, you just have to take my word for it, but we saw a very strong antiviral response in both the honeybees and the mites. So we perform this method called small RNA sequencing, and this allows us to measure how strong the immune response is to the viruses. And the bee response has got this yellow response here, very strong degradation of the virus, and the mite response also has a very strong degradation, but their responses look different. Now that's a story for another talk, but for the purposes of today, all I want you to know is the viruses are present in mites at high frequency, in bees at low frequency, and the immune system is reacting to these viruses in both bees and mites. So that was about 2017 when that came out. Now, let's look a bit closer to home. And so there's been a lot of work looking at honeybee viruses in Australia. So of the common viruses, we have about 5 of them. We don't have some of the worst viruses in the world, so deformed wing virus and slow bee paralysis virus. Now, I say that with a caveat because some of you may have heard a recent update from ARBIC that there has been a detection of deformed wing virus in Australia. Now, this is a very unique identification of deformed wing virus because it was found in these invasive Apis florea, the dwarf honeybees that are so far quarantined, isolated in this Burrup Peninsula in Western Australia, so near Dampier. And these bees have been first identified in Western Australia in 2023, and subsequent testing of stored samples has shown that deformed wing virus was present at low levels in one of about 40 colonies that were tested. Sorry, excuse me. So, while historically we could say that based on extensive surveys and sampling, deformed wing virus was not in Australia, in fact, we do have it, but it's just located not in our western honeybee, but in our invasive Apis florea off the coast off this very peninsula in Western Australia, and has hopefully not spread any further from there as of yet. So my colleague John Roberts from CSIRO performed a really extensive survey across Australia in around 2015. So it's getting on 10 years now since he did this survey, and he surveyed over 1,000 hives from 140 apiaries in every state of the country. Now, John searched for new viruses using that metatranscriptomics technique that I mentioned, and he managed to find 42 novel viruses in Australian bees. Now, I've got a couple of family trees here. I mentioned these positive stranded RNA viruses before. All the viruses that John found were from those positive strand families, and these are the two bee families, the Iflaviridae and the Dicystraviridae. Anything in red are ones that John found in Australia. Some were known already and some were completely new to science. So these are the ones that are labelled with their state name. Now, thankfully, he didn't find deformed wing or slow bee paralysis or some of the other paralysis viruses in blue, but there are certainly quite a variety of new viruses that were present in many different locations in Australia. Now, the question that comes on to everyone's mind is now what's going to happen now that Varroa is in Australia, and what will happen to our viruses? This is one of the main questions that drives my research. So we have these mites. They've arrived in 2022 and they're slowly spreading their way around the country. And we also have a whole set of new viruses that mites potentially have never encountered before. Interestingly, and very luckily, these mites, by chance, did not bring deformed wing virus with them. And so we have this novel scenario where we have a clean slate. We have mites without deformed wing virus, but we also have Australian bees and all their weird and quirky viruses that John has found in his previous work. So there's plenty of things to be asking about what's happening in our bees. So I've just got this little time-lapse video, which is obviously not my video. It's way too fancy for my iPhone to take, but it is, you will soon, hopefully not too soon, down there in Melbourne, but experience the very interesting observation of seeing what mites do in your hives. It's quite amazing to see them live in the flesh. It's also hard not to be afraid of them, but I try and look at them with this sense of wonder rather than fear. So this is how I'm approaching our realization that mites are here to stay, is that they're quite fascinating organisms, and there's lots of research being done to figure out how they work. So basically, we all know this already. They weaken bees by feeding upon them. They damage the bee's cuticle at this pupal stage, and they replicate within the bee comb. And during this process of feeding and reproduction, viruses are passed from bee to bee, so from mite to bee, sorry, and the mite that bites the bee will pass on any viruses that it carries, and also some viruses will increase inside the mite as well. So looking now at how Varroa changes the viral landscape by adding this vector into the system. Previously, viruses used to rely on that bee interaction to spread, so you would need a virus to be spread from bee to bee. Now that there's mites in the picture, they are able to carry viruses, and if viruses replicate in the mite, they can deliver a big dose of virus to a bee, much bigger dose than what a bee would pick up just by contact with another bee. So the mite therefore changes that viral landscape because it changes that whole transmission dynamic, increasing the prevalence of viruses in hives and their viral load. So the main virus that has changed globally since the arrival of Varroa is the deformed wing virus, and lots of correlation studies around the world have shown that when Varroa arrives in a particular location, previously low levels of deformed wing virus will rapidly increase. And if you don't treat the hives for the mites, then it's ultimately the viruses that will cause these hives to collapse. So getting rid of the mites is a pretty effective way of getting rid of the viruses. Now, we've shown this relationship experimentally. So this is just some studies that have shown that it is really that deformed wing virus that is associated with mites. This is based on observations, but a student of mine, Amanda Norton, showed in an experimental setting when she experimentally changed mite loads in the Netherlands. Essentially, she performed a study where she added mites to a set of hives and removed mites by treating them from a different set of hives. And what she saw was when the mites were not present in the hives, we had two viruses that we were looking at, deformed wing virus A and deformed wing virus B. Now, when the mites were not present in the hives, these virus levels were low. And when the mites were added to a different set of hives, the virus levels were increased much higher above that mite removal set of hives. So that putting in of mites, increasing the virus loads is kind of replicating what everyone else has observed throughout natural settings as well. So the experiment has proven that association. So what will happen in Australia without these deformed wing viruses? Now, it could be a case where we have other viruses that we already know about. So Israeli acute, sac-brood virus, black queen cell virus increase because of mites. But throughout the world, what we do find is that these viruses are too pathogenic to bees. So they kill the bees too quickly, and therefore they are not really good for mites, because the mites need the bees to stay alive so they can stay alive themselves. So we're now wondering whether there will be some other virus that can come along and take on that role of deformed wing virus in its absence. Obviously, we could also have another incursion of bees where deformed wing virus gets brought in somewhere else, and then when the virus and mite join together, we will see what has happened throughout the world. But we also have a whole heap of Australian viruses that might be doing some interesting things. Now, I have this project at the moment where I have a student who's sampled throughout the Varroa affected area in Newcastle, Kempsey, and in Sydney. And we had this lovely set of samples where we could get pre and post Varroa from the same hives. And this is Elizabeth here. She was a master's student. And what she found is when screening for a few of the viruses in Australia, that there was a very strong correlation between these two new rhabdoviruses that I mentioned prior to my earlier in my talk, where when you have a high load of mites, so the mite load is increasing along the bottom, you actually get a higher load of the viruses, which is increasing along this increasing upwards. So this correlation, these green and orange lines, is correlating the virus level and the mite level. Whereas other viruses like black queen cell and sac-brood were pretty flat. So that means it doesn't matter how many mites you have, those virus levels were kind of staying the same regardless. So this correlation suggests that mite presence is driving the levels of the Apis rhabdoviruses. So these are the two novel viruses that I mentioned that had spread globally. Interestingly, these viruses had not been found in Australia prior to Varroa being here. So it's possible that these are the two new viruses that are going to be very interesting in Australian bees as mites spread. So I am currently working on a small grant with AgriFutures where I've had some funding to do some transcriptome analysis of these bees and mites in the invasion zone. And I hope to be able to track change in viruses over time and look at how the immune system of bees and mites are responding to these Australian viruses. We are also doing some experimental work looking at how these two rhabdoviruses are actually being transmitted to bees. So, say, taking mites out of a hive and putting them onto little pupae that have never been exposed to mites. We had a lovely experiment where we had some pupae from uninfected hives and mites from obviously infected hives, where we placed these mites onto pupae, and we're looking to see how often they are being transmitted, the viruses are being transmitted. We're trying to confirm that the virus is coming from the mite into the bee and not just simply being transmitted from bee to bee. And we're also trying to figure out some ways to understand what symptoms these rhabdoviruses might be causing in bees, because while we can find viruses using metatranscriptomics, it's just telling us what the virus sequence looks like. It's not telling us what that virus does to bees. So there's possibly some negative impacts of the viruses in our bee population, or they could just be viruses that are simply hanging out and lurking there unnoticed in bees, which is definitely possible. Look, I think I've talked your ear off enough for 9:30 in the morning, but I just wanted to thank you for listening to me remotely. I'm sorry I couldn't be there again, and if there's any questions or any comments, please feel free to ask.